Thank you, Ellen. So I'd like to invite all the speakers from this session to come up front, and we're going to have a panel discussion. We've got about 25 minutes or so for open discussion with the audience, and we certainly would invite questions and comments. If you wish to make a comment, we ask you to go to the center microphone, and or I guess there's a microphone on the side here as well, and identify yourself and your institution. But maybe I can just start off with a question to the panel. It sort of stems from Mark's opening comments about the learning and confirming paradigm. And I guess the question in my mind is, how much of the confirming needs to be done pre-market, and how much of it can be done post-market? And are prospective randomized clinical trials the only confirmatory strategy that's really acceptable? I have to wonder, quite honestly, and maybe Mace and Janet could comment on this as well, whether those two large prospective randomized clinical trials to prove the benefit of crizotinib were really necessary, particularly since the drug was approved by the FDA before the results of those trials were even known. And how many resources were devoted to conducting those trials, and how much more information did they provide about the drug than we had when the drug actually was approved for marketing? So, I think one of the things that we need to consider throughout this workshop is, what needs to be known when, and how confident do we have to be in our knowledge at any given point in time to take a particular action? So let me throw that question to the panel members. And Mark, maybe you want to just give us a quick thought on, in the learn, confirm paradigm, do you think all of that information that you described needs to be obtained in the pre-market setting, or, when can we start transitioning to getting that information post-marketing?
No, it's a great question. And, I think one thing we generally fail to get wrong early and often fail to have right at the time of approval is dose. And so the real issue is, well, do we need to get the dose right at the time of approval? What if we're too high? And I think it comes back to the elephant in the room, which is pricing and costs. And just to put it in context, abiraterone, and not represented on the panel, but it's Janssen's drug. I mean, we conducted a study post-approval without any involvement of the company, looking at the labeled dose, which is 1,000 milligrams fasting versus 250 milligrams with food, a randomized phase 2 trial of about 80 patients, multi-centered. Don Harvey was involved in that study. And the lower dose is non-inferior when you look at a reasonable biomarker, which is PSA change at 12 weeks. Okay, so now, we're not suggesting a label change, and that potentially has a huge impact on revenue of the drug for the sponsor, but it doesn't need to. In other words, if we changed our pricing models so that it was like per patient month or per course or something like that, we wouldn't really care. Companies would feel more comfortable optimizing dose after approval, and the world would be a happier and better place. So I don't know, I don't know what Mace, it'd be interesting to hear Mace's comments on what happens if you don't optimize the dose pre-approval and how much Pfizer cares about that.
Thank you for that question, Mark. So I think that it's a balance. You get one chance at drug development. If you take the wrong dose, too high or too low, into your pivotal trials and fail, there's no second chance. But at the same time, we work in a very competitive environment. So we have to balance these things. I think with regard to food effect studies, those are routinely done pre-marketing. So we need to know what the effect that antacids have, what the effect of food has. Sometimes it will change because the formulation that you use in your early clinical trials may change as you move towards the market. And sometimes that also changes the relationship between your drug exposure and food and gastric pH. So it's very dynamic. You're constantly learning. So I think that that's really important. I think that we have to recognize that when we bring a drug to market, we never have full certainty. We never can have full certainty. The issue is, do we know enough to be able to accurately characterize the benefit-risk relationship for the patients who are intended to get the medicine? And so there's a whole process of post-marketing surveillance, pharmacovigilance, that lead to very regular label updates, usually updating experience that we have in children, in individuals who have liver or renal dysfunction, so we can allow the practicing physician to be able to utilize these medicines appropriately in those rare patient populations. So we also have life cycle management and have some dose finding that could be done there to try and optimize things. But when we look at actually whether one dose or schedule is optimal, you have to ask the question, what would that trial look like? And what would be your endpoint? How long would it take? And when it finally does read out, is it going to be relevant to the way the drug is being used at that point in time? And will the endpoint be sufficient to change clinical practice?
Janet, any comment on this issue of
Sure.
pre-market versus post-market, and how much confirmatory data is really necessary?
Okay, well, if we consider that we all agree, okay, that development of knowledge around investigational drug is a continuum. It might be steep at first and then level off a little bit, but it never stops. We're still learning how to use digoxin, right? There's major changes in how to use it. But the approval decision is viewed as a dichotomous, it's binary, yes, no, okay? So where do we cut that? And so the answer, unfortunately, is it depends, okay? It depends on the clinical situation. Why do people have these right to try laws and so forth? Because, okay, if something comes along and it's shrinking their tumors dramatically, they don't want to be told, we're going to wait till we do all these other trials and have all this information before you have a chance to maybe have your life prolonged, right? So it depends on the clinical situation, including the number of alternative therapies. It depends on, what do you know about the dose and how much toxicity is there to this drug compared to the quantity of benefit. It depends on the quantity of benefit that has been seen. Is it a breakthrough type of thing that's unprecedented, that might be a potential game changer, or is it incremental? If it's incremental, usually you want to have more information because none of these drugs are a free lunch. In other words, there's always some toxicity. So we have tried to, with not the greatest success, but we are trying to establish a benefit-risk framework where we lay out these considerations in a semi-quantitative way, and we've published a little bit on this so that you can see where, no matter where this is taken in the continuum of drug development, hopefully there'll be some stable balance of benefit and harms where the benefits come out ahead. You obviously have to have the basic information about the target patient population, approximate dose, given that we almost never approve the actual best dose, right? So a reasonable dose finding and a safety profile that supports the benefit is still exceeds that potential risk. So I would say it's really hard to say, and we are making this cut point at different times in the drug development path, and I think that's a rational thing to do.
Okay, let's go to Keith.
Keith Flaherty from Mass General. So Ellen and Janet, I'll direct this to you, but Ellen, you're the one who put one line on your one slide about biomarkers and the problem of unvalidated biomarkers. So all biomarkers are sort of aiming to be proto-diagnostics and in their greatest glory become diagnostics. But there's very little ownership of that problem field-wide, and particularly for what you're talking about, which is not like the single biomarker to unleash Mace's drug, but the platform diagnostic, which we have for next-generation sequencing now, but maybe with immune profiling we'll have it in the next, let's say, 3 to 5 years. go to NGS. So in the NCI match trial, which is, made my entire head of hair gray, which you can't possibly tell that that's true, but it's true over the past 3.5 years, we've done one very powerful thing, which kind of leads me to a directed question about who owns this problem. In the NCI match, we had an RFA for 4 labs to come in and compete to be sequencing centers using the same platform. And then after they were, put in their suggested capabilities, we then had proficiency testing across the laboratories, test sets going around. We did this with CDRH. In other words, without their requesting it, we opened a dialogue and said, look, we think this would be an important thing to do for NGS to have proficiency testing. We have a 5th laboratory actually coming on now. And at the tail end of match, which is about to sunset very soon, we have 2 for-profit laboratories who've offered to come in and essentially do the same thing. In other words, for us to be able to use their platform, at least for a subset of our patients. It's been a beautiful thing, but NCI Match will be gone in 6 months, and this first version that took a handful of academic labs, a couple commercial labs to try to address some of your concerns. That we have the data already, we just haven't published it, we presented it. Very, very high concordance across these laboratories, at least using one platform, looking at hundreds of genes. So when this ends, who picks it up? What stakeholder in our community is the one that can do this work that I would argue is pre-competitive and not the territory of certainly a therapeutic company, and I would argue diagnostic companies, until Janet answers the question, leave her aside for a moment, until there's regulation of biomarkers, someone's got to generate the evidence.
Multiple components to your question. First of all, kudos for you for that NCI for match. It's a great trial. And also, you did the right thing. You had concordance, you had standards. You just didn't go out there and say, okay, 4 centers do it. You made sure before. This doesn't happen in the real world. Right now, you go to MSK or to Stanford or MD Anderson. They have their platform. I'm sure it's pretty good. They say it's pretty good. But I'm a little bit like Harry Truman, show me, prove it. And because this really impacts the patient and clinical decisions. So I think that area of what you did is an example. I don't think anybody is suggesting that every single center has to do their own IDE, but there has to be some standardization. What happens to match after, I don't know. But I can say this is an example of NCI and the academics doing the right thing, and you should be very proud of it.
Well, I think there's been a recognition that the IDE route for every single group may not be a viable pathway. And so there, of course, have been a number of discussions about having a data bank, registering the findings, and that CDRH has been working with the NIH on. But I think it is a problem in the lack of ownership, and we have seen this, the next generation sequencing, like it sounds like from your data and others, in many hands, it's pretty reliable, and CDRH is taking the approach of kind of approving the platforms, and then each single assay could be posted and so forth. However, there are going to be many other, as you alluded to, types of assays that are used in making decisions about patients. Many of them haven't reached the level of maturity that sequencing has. And I think we're going to see kind of what we saw for the immunofluorescence for HER2, that we actually patients are will be disadvantaged by the fact that people do not own these assays. There isn't the kind of round-robin concordance and proficiency that needs to happen. Nobody establishes reference standards and other kind of things that are needed. And it's a huge problem, but there is not an entity that owns this problem. You're right.
Point, and I'll go on. I agree fully. It's unfortunate that the guidance, which was scheduled to come out, did not come out. I think it would have helped us. But I think that CAP and the pathologists and FDA have to work together on this to get some sort of concordance in standards, because it's really going to hurt everyone if we don't.
Yeah, I just want to comment. I mean, the molecular pathology community has focused really on technical issues. But we know that those aren't the only issues. Okay, for example, somebody's In a trial, you might have a mixture of samples collected at diagnosis, from a primary, from metastasis, after treatment, et cetera. And of course, that we don't, as clinicians, we don't expect them all to be the same or yield all the same information. And so I think that we really do have to move beyond the CAP and CLIA, and there are many, many companies out there selling diagnostic tests from CLIA-approved laboratories that have little or no clinical validity, and payers are paying for them.
Let's go to Suzanne.
Switching topics a little bit, but the pace of drug development, I think, depends in large part on what happens internally in the companies that are developing those drugs. And Mace mentioned that typically in pharma there's an early development group and a late development group. So I'd just like to ask you, Mace, how would you see that changing to accommodate seamless drug development?
Great question, but a delicate one, shall we say. One of the things when I made the transition after 20 years in academia to industry that I was attracted to is the fact that I'd be working within an environment where everybody was focused on the same thing, and that was bringing new, important, transformational therapies to the patient. As I've been there for 8 years, I realized that that is true from a high level, but how you measure success is defined in different ways for different groups. So if you're in a company where there is real question as to, well, how productive are your research laboratories? Should we simply just shut them all down and out-license it to small companies, in-licensing compounds, or is this really been productive? You will find a company that is trying to really focus on new molecular entities coming out of the laboratory into clinical testing. That will be their way of responding to that challenge. You go down the road a few years and you say, oh yes, okay, you've been productive in getting these molecules out, but how many of them are working? Then you'll find a different gain to that control to say, boy, if only 10% that we're getting to the clinic are working, we better have a little bit more of a stronger filter on preventing some from making it into clinical testing and failing there. How quickly you traverse that first in human to optimal dose is another measure of success. But if you spend a little more time to refine the dose, to refine the schedule, could you actually get a little bit more, or maybe a lot more out of that? Can you understand how it might combine with other agents? That takes time, but that could be viewed as falling short of certain metrics for success within the company. So I think that it really is incumbent upon us to make sure that we see things from end to end, that we coordinate, and sometimes it's different organizations, and there's not necessarily frequent interactions between those, that we create those. We reach across those organizational silos, if you will, to make sure that we are getting the drugs that we see from our perspective are going to be meaningful to the external community. Being in more late-stage development now, we interact extensively with outside experts like you, like many others here, whereas those individuals, those colleagues who are working in early development, not so much. They're really focusing on, do we have a drug? Can we give it safely? Can we identify an MTD? So I think that there's a little bit of that disconnect within companies and that handoff that we can try and have more formal and more extensive discussions so that we make sure that the drugs that we're getting from the groups, from the laboratory, from early development, are drugs that are going to be meaningful and be successful.
Go to the center.
Yes, hi, I'm Judy Keene with the American Society for Radiation Oncology. You've talked about drug-drug combinations and kind of how those two together in new trial designs. I was wondering your thoughts on combinations that include drugs and radiation and in these new trial designs, and is this something else that we have to consider when you have those type of combinations?
Direct that towards.
I can, Janet? Dr. Woodcock?
Sure. Well, I think I use the word interventions, or I should have, because I think that, for example, when you think about a master protocol or others, there's one stream where you're looking at the drug and you're trying to establish its dose and its activity and its toxicity and so forth. But there's a whole nother endeavor, which is where does it fit into the armamentarium and into the regimen where you're treating that cancer at that stage. And there you really need to optimize with radiation therapy or whatever is going on for the patient. And I think that part isn't very well worked out as far as how that is. Obviously, if those patients need radiation, that will be incorporated into the development program, but I think it's it's an add-on type of design usually, because you don't want to change two variables at once. And that's true for the other chemotherapy agents that the people may be using as well. Often the designs are add-on or compare or subtract one and swap in another, right? But that actually may not be the optimal way to use all those drugs together. So there's more challenges beyond simply the drug development challenge if you think about treating the patient with the disease and treating the disease, and certainly where and how to use radiation therapy is part of that, I think.
I mean, I think conceptually it's actually quite straightforward. I think in practice, my own limited experience is the toughest thing is getting the radiation therapist to standardize anything. And so you have that whole variable that makes it virtually impossible to do a trial because, particularly if you're giving a drug that potentially can, whose toxicities are affected by the radiation, you have to standardize the radiation. And that's really what's been, I think, a major problem for at least many medical oncologists.
Let's go over here. Please introduce yourself.
In the Alliance for Clinical Research Excellence and Safety. One of the threads that's running through everyone's discussion is a combination of collaboration and outreach to different stakeholders, and in essence, culture change. And so one of the questions that I have is, what are the best practices, if you will, that you found that enable collaboration, enable crossing through stakeholder silos, and who pays for it?
Well, if we don't do it, everybody pays for it. So I think there's an enormous necessity to do it. It's hard because it's a change of practice. Before this, everybody did their own thing individually, and I think the complexity of the science and new models that emerged started to say, wait, we better start to do this. I mean, the FNIH has always done public-private partnerships on pre-competitive science, and there was a lot of participation where there was a need. I think the need brings us closer to it. I know, as an example, with LungMap, there was a compelling need to do this master protocol on genomic testing. There, the issue was the very rare populations, which you'd never get to, ever, 2%, 5%, 8%, very small. But it was also the idea that the FDA was willing to make this into a registration trial, which made it very attractive for the companies. And there was something in this for everyone. The NCI was a catalyst, the cooperative groups, the patient groups, everyone came together, the companies, and there were lessons learned. It's hard because when everyone is used to doing things on their own way, and all of a sudden you have a partner, it is really hard. It is really hard, but it happens out of necessity, and if we don't do this, we're not going to get there. So I believe strongly that necessity is going to drive us to these places, and everyone pays and everyone does what they can.
The center.
Okay, thank you. So I was curious to hear from all of the
Would you introduce
stakeholders.
yourself, please?
Hi, I'm Mary Redmond. I'm the LungMap statistician at Fred Hutch in Seattle. So I'm curious to hear from all of you, your perspectives on the way that we're developing the anti-PD-1 and PD-L1 drugs right now, where we have, I think Gideon just told me 22 different inhibitors out there. So if we want to talk about, you could ask me to design the most efficient trial, but if it's being repeated 22 to 800 times, what do we need to do to try to encourage asking the question more efficiently with across different companies?
Take that, and perhaps Mace and others, because you were at the meetings. I think FDA is very concerned about this. I mean, there are 4 or 5 approved. We have a huge pipeline. There isn't enough patient expertise or the ability to even put these on patients on the trial. So what was proposed is some sort of master protocol for some of these newer companies to try to really work towards a registration trial. But candidly, it wasn't well received. There are those that are more advanced and frankly don't have the incentive, and others somehow didn't get there. And maybe, Mace, I don't want to put you on the spot here, but I am, because I know this is a very big concern from FDA.
So thank you for that question. So I think we have to recognize that, with all the decades of experience we have in therapeutic development in oncology, we have never faced a situation like we face today with PD-1, PD-L1 inhibitors. And what I mean by that is never before have so many sponsors gone forward with their unique inhibitors at the same time as with checkpoint inhibitors. Why didn't it happen with a taxane? Why didn't it happen with angiogenesis inhibitors? It's because the potential is considered to be so great that never before had agents been able to demonstrate such remarkable activity in patients with advanced disease. And so there is a great opportunity there. But even so, is that market large enough for 20? I mean, gosh, even hypercholesterolemia only had 4 or 5. There's really 20. And it goes beyond just the use as a single agent. So the thinking is that if you, as a sponsor, don't have a checkpoint inhibitor in your own portfolio, and you have maybe other agents that might combine with that, let's say one of these really does show it really does make a difference and it's better than the single agent. How do you price that? Because you only control the price for your part of that. And then the partner controls the price for the checkpoint inhibitor. So actually, as it was described a few years ago, companies would be divided into 2 groups, those who have checkpoint inhibitors and those who do not, the haves and the have-nots. And so, we thought about this very carefully, and actually, not just because of that, but because it was a good opportunity to share expertise from both companies, went into a partnership arrangement with Merck Serono to develop avelumab. And it really now provides us with a platform for those combinations with other agents that we draw from our own portfolios. And then, if you then have something that really does offer more than the single agent, you can actually work with payers, work with health authorities to be able to find that point where the benefit and the value and the cost all align. When you try and do that across companies, it becomes much more difficult.
Well, I'll put out a controversial concept. So
It's so unlike you.
what if the FDA took the lead and said, we invite any company with a checkpoint inhibitor to participate in a trial that will demonstrate for disease X that their drug is as good as some other drug. And if you meet that bar, all drugs in the trial will get all indications for all other drugs in that trial. In other words, FDA took the lead and basically said, we're going to basically create an incentive for companies to participate so that they can get broad indications. And companies, of course, run the risk that their drug would prove that it's not the real thing. And that it would be an interesting concept. Of course, FDA would have to be willing to give approvals with less than pristine data, but I think it would definitely get over some of the roadblocks we have. See Rick in the back.
Can I comment on that? Actually, we have done this, and we have proposed such designs. And of course, it's ultimately left to the sponsors to take it or leave it. We can't force them.
Okay.
We partnered a meeting with the Sean Parker Foundation and many of the companies and investigators on precisely this area. And some of the smaller companies were somewhat interested, the larger companies not so much, and I think maybe for some of the reasons Mace are suggesting, but these were registration pathways.
But I guess what I'm saying is if a larger company weren't interested, the trial could be done without them because their drug's approved and it can be obtained, and the trial can be done with or without the support of a sponsor. They just wouldn't stand to get the benefit of agreeing to participate, but their drug still can be used as a comparator.
It's a little
Harder to finance and do. It's more of a comparative effectiveness. I think what's being proposed by FDA is different, and maybe later we can discuss the nuances. It's a very different concept, but it does get these drugs to market, and obviously everyone's worried about the combination.
So we're going to have to bring this session to a close, but just as a concluding remark, I mean, I would say that what we've already surfaced, in a sense, that everybody's well aware of, is that one of the real challenges that we consistently face is aligning the culture of drug development with the science of drug development. As Janet pointed out, the science is typically far out ahead of the cultural change that's necessary. To some extent, the things that seem to drive changes to the culture, ultimately come down to what is the opportunity, both the medical opportunity and the business opportunity. What's the competition? What's the landscape look like? And what's the risk tolerance of the sponsor? How willing are people to fail at various points in the process, and what are the consequences of doing so? So hopefully we'll have an opportunity to explore these issues in further detail in the remainder of the workshop. We're scheduled now for a coffee break. Let's plan to be back in your seats by no later than 10:20, and then we'll go on to the next session. Thanks.